Okay, what are we going to do today? We're going to talk about lymphoid tissues and how they're strategically placed to effectively protect us from any number of infections by pathogens. And we're going to walk our way through this in three main categories. First, we're going to talk about the spleen. We talked about the spleen yesterday, but yesterday's discussion of the spleen was in terms of the sites where the red pulp would be a mixture of the macrophages and the worn out red cells, worn out erythrocytes. Today we're going to think about something a little bit different. We're going to be thinking about the white pulp, and the white pulp will be cells of the immune system. Now again, we're going to exploit the ability of cells to get out of the blood vascular compartment and into the spleen. Yesterday that exploitation was through the erythrocytes, and today the exploitation will be largely through bacteria and other pathogens which can leave the blood vascular compartment and get into the spleen, where they're now going to be cells of the immune system. The cells of the immune system then can attack those pathogens. The next type of lymphoid tissue we're going to talk about are the tonsils and Peyer's patches, and these are sometimes known as the gut-associated lymphoid tissues. They're found all along the GI tract. The ones we're most familiar with are the tonsils, because we can see them in the back of our throat. If you've ever had a sore throat and you Okay, to try again. So let's start here with the tonsils and Peyer's patches. I think we're okay with the spleen. Tonsils and Peyer's patches are sometimes known as the gut-associated lymphoid tissues because they're associated with the epithelium of the GI tract. The place where you can see them is you can see them in the back of your throat. And if you have tonsillitis, you can open your mouth, look in a mirror, and you can see those inflamed tonsils. And what's going on there is you have a bacterial infection in those tonsils, and the tonsils are now trying to mount an immune response. And we'll talk a little bit about how that gets done. But this is going to be one of the major players in your immune system. Then the third set will be the lymph and lymph nodes. Now, we talked a little bit about lymph the other day when we talked about hemodynamics. And again, we're going to emphasize this idea of hemodynamics, where there is a greater pressure on the walls of the capillaries. That's the mechanical pressure, the hydrostatic pressure. That's greater than the osmotic pressure, bringing the fluid back in again. So there's a net flow. There's a net flow of fluid out of the capillaries into the extracellular space. That fluid now in the extracellular space needs to be drained. And that fluid is now drained through a series of tubes and vessels called the lymphatic vessels. And there are a whole series of these vessels throughout your body, and they will extend then back into the subclavian vein. So there's a kind of circuit. The fluid gets extruded out of the capillaries, gets collected in the tissues, and then that collected fluid is then passed into the lymph vessels, and they then pass through the lymph nodes. Now we're going to see in the lymph nodes, again, we're going to have clusters of cells of the immune system, and those clustered cells of the immune system now provide a line of defense, because this lymph will have been pushed out of the blood vascular compartment. Now it's going to bathe tissues, all of the tissues. It doesn't appear in the brain. We don't have lymph in the brain, but in all of our other tissues we have this lymph production. And the lymph production then provides a fluid which bathes all of our cells. And then any pathogens which are in that extracellular environment around those cells can be picked up by the lymph and moved to the lymph nodes. And then within the lymph nodes there are cells of the immune system, so we can at that point expose our immune system to those potential pathogens. And the fourth character is something entirely different. The fourth lymphoid tissue is called the thymus. The thymus is the structure, it's basically in your upper thoracic cavity around your throat. And the thymus plays a really, really important role, mostly in the early stages of development. You probably at your age still have a fairly active thymus. Mostly it's active when you're a child, and as you get older and older, the thymus begins to wane in its effects. But what the thymus does is it is dedicated to T lymphocyte selection. We're going to see that there are types of cells in the immune system, particularly in the adaptive immune system, called T lymphocytes. All of those T lymphocytes have to pass through the thymus in order to be functional. In fact, they get their name because they have to pass through the thymus. And in the thymus, those T lymphocytes are now selected for both positively and negatively. That is, they better be functional, but they better not recognize their own proteins as foreign, and we'll walk our way through how that gets done. Then we'll drill down to talking about types of immunity, and there are two broad categories here, one of which is innate immunity, sometimes known as nonspecific immunity, and the other is adaptive immunity, sometimes known as specific immunity. When we think about the adaptive immune system, we break it down into these two big categories. One is going to be called the cellular branch. The cellular branch is dependent upon T lymphocytes. It's dependent upon those cells of the immune system that have been selected in the thymus. And then the other branch is the humoral branch. And the humoral branch is predicated on B lymphocytes. So you'll need to keep these two cell types separate, T lymphocytes and B lymphocytes. Then we'll talk a little bit about the characteristics of these different types of immunity. When we talk about innate immunity, what we immediately think of is a kind of holding action. What the innate immune system is doing is it's trying to prevent your entire body from being overwhelmed by pathogens. And it's doing that in such a way that it's rapid, it's not very targeted, and it's potentially destructive. And we'll talk about this in terms of both viruses in the form of interferon and bacteria in the form of inflammation. Now, adaptive immunity, in contrast to innate immunity, is slow. It takes 10 days to 2 weeks to really come on in full force. And the reason it's so slow is that it initially begins with a single cell. And that single cell then has to proliferate and make antibodies, whether it's really going to be part of the T cell system or the B cell system. But the adaptive immune system is highly targeted. It will generate antibodies or T cell receptors that recognize short segments of amino acids and potentially pathogenic proteins. Segments of amino acids that are like 16 to 20 amino acids long. And the other characteristic of the adaptive immune system, which is clearly different from the innate immune system, is that it has memory. It has a mechanism for recognizing pathogens that it's seen before. Whereas for the innate immune system, every introduction to a pathogen is a brand new event, and it does the same thing over and over. And then we'll drill down and talk about these two kinds of immunity in terms of interferon for viruses and bacteria and inflammation. So let's start here, though, with a list. And we're going to start with lymphoid tissues. And the first of the lymphoid tissues we're going to talk about is going to be the spleen. And the spleen is effective in monitoring for blood-borne pathogens. And in fact, when a spleen is removed from a child, you find that those individuals for the rest of their lives are more susceptible to blood-borne pathogens. For the most part, if your spleen is removed later on, you have backup systems. But the spleen is monitoring for blood-borne pathogens, and this is then predicated on the idea of the fenestrated endothelium. Just as we saw yesterday that the fenestrated endothelium permitted the movement of erythrocytes from the blood vascular compartment out into the extracellular regions of the spleen, we can use this same trick, these wide gaps between endothelial cells, to allow for the movement of bacteria out of the blood vascular compartment and out into the extracellular space. And then in the extracellular space, we're going to find regions known as the white pulp. The white pulp is, in fact, something called a germinal region. For cells of the immune system. We're going to see this is a common sort of theme for all of these lymphoid tissues except for the thymus. The next lymphoid tissue we'll want to talk about are the tonsils and Peyer's patches. Sometimes you'll generally see these referred to as gut-associated lymphoid tissues. More characteristically, they're associated with the epithelium of the GI tract. And we'll see that what they're doing is they're monitoring for pathogens on food, in water, or in air. And you can see how these are strategically located. If you think about your tonsils in the back of your throat, those, every time you breathe air in, your tonsils are now exposed to that breath of air. Every time you take a swallow of water, every time you take a swallow of food, those structures are exposed to potential pathogens from the outside world. So they're monitoring for pathogens from the outside world. And again, we'll see that they will have a germinal region. It's going to be a germinal region beneath the epithelium. To monitor for pathogens. The third type of lymphoid tissue is going to be lymph and the lymph nodes. Lymph is fluid that is pushed out of. It's pushed out of the blood vascular compartment and returns to blood via lymphatic vessels. And then all along that pathway, all along those series of vessels carrying the lymph, there will be these structures called lymph nodes. And the lymph nodes contain germinal regions. Again, with cells of the immune system. The fourth lymphatic tissue is going to be the thymus. And the thymus has an entirely different function. The thymus monitors newly generated T lymphocytes. All of the cells of the immune system, all of the erythrocytes, all of them originate in the red bone marrow. And that's true for lymphocytes as well. They're born in the red bone marrow, but they have to mature and be selected in the thymus. So we're going to see that these newly generated T lymphocytes leave the red bone marrow and migrate to the thymus. And in the thymus, T cells, or T lymphocytes, are selected for efficacy and potency. And we're going to see that when we talk about this, this is said to be positive selection. And the positive selection is, can the lymphocyte recognize protein? Let me say that a little differently. Recognize pathogens. But the negative selection is basically can or does the lymphocyte recognize self-proteins. If you generate lymphocytes, particularly cells whose destiny will be to kill other cells, kill virus-infected cells, potentially kill cancer cells, if you have these T lymphocytes going around randomly in the body, recognize your own proteins and killing your own cells, that's bad. That's an autoimmune disease. So one of the things the thymus does is it selects for T lymphocytes that can recognize foreign proteins but don't recognize our own. So those are the four main characters that we're going to talk about in terms of the lymphoid tissues. So let's start here with the spleen, and you'll recognize a lot of what we talked about yesterday. So this is meant to be the spleen. There is an enormous blood supply into the spleen through the splenic artery. And commonly after an accident or after a bad fall, there's a rupture of the spleen and the blood supply gets disrupted and you begin to hemorrhage from that spleen. Now, if that isn't repaired surgically within a matter of 24 hours, you can be in some serious trouble. So it's not uncommon for people to have their spleens removed after they've damaged them. But nonetheless, the spleen, because of its heavy vasculature, is drained by a splenic vein. That's all familiar from what we talked about yesterday. And again, familiar from yesterday is there will be a series of capillaries. And those capillaries are unusual in the spleen in that the gaps between adjacent endothelial cells are large. In most capillaries, the endothelial cells are very close to one another and don't let large molecules pass. We'll talk about that when we talk about lymph. We mentioned that idea when we talked about hemodynamics. But here, the endothelial cells are widely spaced. Yesterday's story told us that those worn-out erythrocytes could leave the capillary and get out into the extracellular space, into the interstitial space of the spleen. And that we said yesterday that there were regions where we could cluster macrophages. So we would see macrophages and erythrocytes. And those were the sites called the red pulp. And the red pulp is the site of the destruction of worn-out erythrocytes. But in addition to the red pulp, there are going to be regions in here called white pulp. And the white pulp is basically a germinal region. It's a germinal region where cells of the immune system aggregate. Now that becomes important because the fenestrated capillaries in the spleen will allow for bacteria, which are only a fraction the size of erythrocytes. They will allow bacteria to escape the blood vascular compartment and come out into the interstitial space. Now, as these bacteria leave the capillary, they're exposed to cells of the immune system. They're being exposed to cells of immunity in the so-called white pulp. So we can think about the spleen then as being a site where we can monitor for these blood-borne pathogens, because the blood-borne pathogens, potentially bacteria, can leave the blood vascular compartment, get out into the interstitial space, and in the interstitial space, these bacteria now can encounter cells of the immune system. And by encountering cells of the immune system, they can then be basically killed and controlled. In many ways, much like the destruction of the erythrocytes that were worn out in the red pulp. Yes?
Is there only one white pulp, or are there like lots of?
Oh, is there only one? No, I make one circle here and say that's white pulp, but there'll be many, many of these throughout. And we'll even see in the lymph nodes that there will be several of these germinal regions within a given lymph node. There will be probably hundreds or thousands of these clusters of immune cells in the spleen. So there's not just one. Yeah?
What do you mean by germinal region?
What do I mean by a germinal region? Let me define that. A germinal region is a site in lymphoid tissues where cells of the immune system aggregate. Now, one of the reasons they're called germinal is that among the immune cells you'll have in here are what they're called naive T cells or naive B cells. And these are cells that have freshly come out of the red bone marrow, and they're waiting to encounter some foreign protein, some pathogen. And then they're germinal because then there will be signals, and we'll talk in some detail about these signals, that will tell these cells of the immune system to begin dividing and proliferating and make copies of themselves and making thousands and thousands of copies of antibodies that will be specific for antigens carried by the pathogens. Okay, so, but when we talk about germinal regions, think about sites where cells of the immune system cluster. And then those are strategically located here in the case of the spleen, so that if we find some sort of bloodborne pathogen that we've never seen before, then those cells in the germinal region can then proliferate through a whole long series of communications from one cell to the next. They can start to proliferate and make copies of themselves to directly attack those pathogens. But we'll call them germinal regions because that's where the immune system largely germinates. And we also have memory cells stored there too. We'll talk about that. Anything else? Okay, so that's the story of the spleen. And let's now talk a little bit about the next set, which are going to be the tonsils and Peyer's patches. Sometimes known as the gut-associated lymphoid tissues. Now, what separates these from the other types of lymphoid tissues is that they are associated with the epithelial cells. Associated with the epithelial cells of the GI tract. The ones we're most familiar with are the tonsils, but you'll find these clusters of immune cells all along the gut. You find them in the small intestine. You don't find them so much in the stomach, but you find them all along the small intestine and into the large intestine. And if we look at these, there are some characteristics of them. So here's going to be an epithelium. This is the epithelium of the tonsils or Peyer's patches. And it's a classic epithelium in that this is a surface epithelium. And the surface epithelium is thrown into these invaginations. Now, we talked about these in glands, but this is not quite the same thing. But this structure in the tonsils and Peyer's patches gets a name. It's called a crypt. And what we do, and we do this deliberately, particularly when we're younger and we're trying to build an immune response, we'll actually capture bacteria in here. Streptococcal bacteria, staphylococcal bacteria will get trapped in these crypts. And right on the other side of that epithelium, there's a region in here called a germinal region. And this germinal region, as we've said before, contains cells of the immune system. And when these bacteria are caught in these crypts, they're giving off proteins. Proteins are sloughing off. And there are specialized cells among the epithelial cells have this name called M cells. And the function of the M cells is to transport proteins, in this case, potentially pathogenic proteins across the epithelium. So basically, this is typical endocytic, exocytic pathway, where these proteins will be taken up by endocytosis, passed into vesicles. They're taken up on the apical side, packaged into vesicles, and then those vesicles are released on the basolateral side. And these proteins now can find their way to the germinal region where the cells of the immune system can respond to those potentially pathogenic and foreign proteins. So the tonsils and Peyer's patches all along your gut are an important part of the immune system. And I think you can see that because they're located along the epithelium of the gut, they're going to be monitoring for pathogens you take in with everything you eat and drink. And the ones in the pharynx, up around your throat, those can be monitoring for pathogens you take in through the air. And so these potential pathogens get stuck in these crypts, and then we deliberately move proteins of the bacteria across the M cells and expose the immune system to those cells so we can mount a proper immune response. Now, we recognize in our modern world that the tonsils are an important part of our immune system. Now, when I was a kid, most children had their tonsils removed. They basically got to the age of 7 and they took their tonsils out, didn't think they were of any use anymore. And that's certainly not true. But now, the only time you see tonsils being removed are among people who have had chronic and repetitive cases of tonsillitis. And we think what's happened in those individuals is they have gotten some form of bacterium caught in the crypts. And then that bacterium continues to produce proteins, and they'll throw off that bacterium, but they never get rid of it entirely. The immune system never quite conquers the bacterium. So these bacteria take up housekeeping in the crypts. And then the individual becomes stressed, they become immune compromised, something happens, and these cells start to divide again. And you get tonsillitis, you get inflammation in the tonsils. In those individuals, it's a pretty good bet that by removing the tonsils, you can stop this recurrent event of tonsillitis. That seems to work pretty well. But for the most part, people in this modern day and age don't have their tonsils removed unless they have these recurrent events of tonsillitis. But it is an important part of the immune system. And one of the ideas that's prevalent here is that what we're doing during childhood, one of the important aspects of the immune system in childhood, is to mount an immune response. And so when you're in your early days of childhood, you're constantly testing out various pathogens to mount an immune response to it. And it's dangerous, and many children, until about 100 years ago, didn't survive childhood. But in fact, it's a way the immune system sort of teaches itself to deal with these foreign pathogens. And as we get older and older, this ability to recognize new pathogens and respond to them efficiently gets worse and worse. So if you're going to get sick, mostly do it when you're a kid. Yes?
What is it like on that like little structure?
I'm sorry?
What is it on that structure that would like pull first? Like as you grow up, like what is it like the M cells that like can no longer take it?
Oh, so you're asking how we get the proteins across the epithelium?
I'm just curious, so as you get older, what is it that kind of deteriorates?
Oh, okay. As you get older, what deteriorates? One of the things that will deteriorate is something I'm going to talk about in a few minutes, and that's the thymus. And the thymus is where you generate T cells. And the T cells will be both T killer cells and T helper cells. And we'll talk about this. But the T helper cells are important in getting other immune cells to proliferate and make copies of themselves. And so one of the things your thymus does is early on in your first 15 years or so is it's generating a lot of these T lymphocytes so that you can then recognize new pathogens. And I don't know, I've never seen this written down, but my suspicion is one of the problems with the difficulties that older people have with COVID is that they don't have the plastic immune system they had when they were 15. And it's one of the reasons why young children are not getting normally very sick with COVID, because they still have this plastic immune system. So they see the virus and immediately their plastic immune system says, oh, I can adapt to that. And they generate antibodies and they generate T cells that can destroy that virus. But as we get older, that thymic system, the system of T cells is no longer as plastic. And it's no longer able, we don't have as many of them, and it's no longer as capable of saying, oh, here's a new T cell, send it out into the world, and recognize these new pathogens. And I think one of the susceptibilities you see in people, older people particularly, for the COVID-2 virus, is that they no longer have this plasticity of the immune system. So they can't mount a really good immune response. It's one of the reasons, too, that we're going to be seeing arguments here coming in the next few weeks about whether or not people in a certain age category, beyond maybe age 50 or so, should be getting booster shots for the virus. And so we'll see how all this goes. But all of this comes back to the fact that as our immune systems age, when we encounter a novel pathogen, we just don't respond to it as well as we did when we were kids. And so I think the answer to why things age, it's mostly in here in the cells in the germinal region, that they become not so effective anymore. Yes?
So are the Payer's patches located directly throughout the GI tract?
The Payer's patches are located throughout the GI tract. The tonsils and adenoids are found in the pharynx. But then as you get down, you don't see many along the esophagus, you don't see many, as far as I know, in the stomach. But once you get into the small intestine, into the duodenum and the jejunum, and I think even as far as the ileum, then you see these clusters of these sites. And again, it's not so bad now. We have pretty good public health measures. The number of bacteria you take in every time you eat something is way smaller than it was 200 years ago. And so there was a time when we needed a strong immune response to all of these things that we ate and drank. Anything else? So this is the gut-associated lymphoid tissues. The next character, and I'll erase this and go over to the right-hand side of the board. The next character is going to be lymph and lymph nodes. And we touched on this. We talked about the generation of lymph when we talked about hemodynamics and the capillaries. Remember, those capillaries are only a single cell layer thick. And because they're exchange vessels, one of the things that's going to happen here is that fluid is going to move from the blood vascular compartment into the extracellular space. Now, I want to emphasize this, not very much. It's a tiny, tiny fraction of the volume in the blood vascular system that leaves, but there is a small amount that leaves. And so we'll start this with the concept of hemodynamics. Hemodynamics and the generation. Of lymph. So again, to remind you, we have these endothelial cells. So these are the endothelial cells. I'll just label them as capillary endothelial cells. And that if we imagine the blood flow is from left to right, and if we remember that we can trace the blood here in the capillary back to the arterioles, back to the arteries, back to the left side of the heart, we recognize that there remains a sizable pressure out here in the capillaries. Maybe, 15, 20, sometimes as high as 50 millimeters of mercury. So there's going to be something known as a hydrostatic pressure. Maybe the best analogy I can think of for a hydrostatic pressure is this mechanical pressure, but it's of fluid. And if you imagine blowing up a balloon, you're generating a kind of hydrostatic pressure, an outward pressure against the walls of the balloon. Well, the heart, because it's pumping, is generating a pressure all along those series of tubes. And there's a pressure here against the tubes called the hydrostatic pressure. That hydrostatic pressure is moving water and ions and small molecules out of the blood vascular compartment out into the so-called interstitial space. But it turns out that the gaps between the endothelial cells are so small, they're kind of like a sieve. The gaps between the endothelial cells don't permit the movement of plasma proteins. Remember, we said that the blood plasma was basically a protein solution. Well, these proteins typically have a molecular weight of about 40,000. And they're just too large to move between the gaps that exist between the endothelial cells. So the mechanical pressure, the hydrostatic pressure, is moving fluid out of the blood vascular compartment. But now the concentration of proteins inside the tube is beginning to increase. And as it increases now, there's now an osmotic pressure. To return fluid to the capillary. So you have these two forces working against one another. The outward force of the hydrostatic pressure and the inward force of the osmotic pressure. Now the hydrostatic pressure, we'll say H, hydrostatic pressure, is greater than the osmotic pressure. Well, if the hydrostatic pressure is greater than the osmotic pressure, we're going to get a flow. So there's a net movement of fluid from the blood vascular compartment, from the capillary, out into the extracellular space. And this fluid, now, the fluid in the extracellular or interstitial space, is called lymph. I think you can see that if you didn't have some means of draining this fluid, that the fluid will build up in these sites. And so there are another set of vessels in here, also lined with an endothelium. And these are lymphatic vessels. They're separate from the cardiovascular system. They're a different set of tubes. And out here, along with these capillaries of the blood capillaries, there are these lymphatic capillaries. These lymphatic capillaries will drain into lymphatic vessels and in lymphatic trunks, but eventually this fluid is returned to the subclavian vein. So there's a kind of circuit if things are working normally. Fluid leaves the blood vascular compartment, goes out into the tissues, and all tissues except your brain. Those fluids are collected in the lymphatic vessels and returned back to the blood vascular compartment in the subclavian vein. So it's a circuit. Now, compared to the amount of blood that's moving through the blood vascular compartment, this is tiny. It's a liter or two a day. But nonetheless, what we're doing is we're forcing fluid out of the blood vascular compartment, and then that all of the cells and all of our tissues now can be bathed with this fluid. And that fluid is now collected in these tubes and returned back to the subclavian vein. But along the pathway of the return of the lymph are these structures known as lymph nodes. And the lymph nodes have this sort of generalized structure, like a little bit like a kidney. And they have vessels flowing into them. And these are the so-called afferent lymphatic vessels. And all along the return pathway of those lymphatic vessels, coming from the capillaries back to the larger vessels to the trunks, all along that pathway we'll find distributed these lymph nodes. On the other side here will be another set of tubes called the efferent lymphatic vessels. So now that fluid, which was pushed out of the blood vascular compartment, which has now bathed all of the tissues, that fluid now is moving through the lymph nodes and back out again. And someplace farther down the line will be another lymph node. But you can, I think, see where we're headed with this. There are going to be within the lymph nodes these structures called the germinal regions. And again, the germinal regions are sites where cells of the immune system cluster. So what we see here is the monitoring of all of the fluids throughout the body, because the lymph is going to bathe all of these different cells, and it's now going to be moving through these lymph nodes and across the germinal regions. This is now a monitoring system for all fluids throughout the body. So whether it's in your lungs or your liver, whether it's in muscle, you're now going to be having those fluids that have passed out of the blood vascular compartment move through the extracellular space. And as they move through the extracellular space, they could potentially pick up pathogens. There's an old story here, and many of you may be familiar with it. It's called the swollen lymph nodes were called buboes. In the mid part of the 14th century, about 1347 to 1352, there was the bubonic plague. And the bubonic plague was known for the buboes. And what happened in this particular case is the bacterium, Yersinia pestis, would get into the body one way or another, usually through a flea bite. And then those bacteria would collect in the lymph nodes and cause a swelling of the lymph nodes. And as the lymph nodes would swell, particularly in the armpits and the inguinal areas between your leg and your torso, then you would get these swollen lymph nodes, and they were called buboes, and then they would turn black, and then people would die. And it killed somewhere between a third and a half the population of Europe. It probably also killed sizable proportions of the population of China, because it originated around the Black Sea and then moved from the Black Sea through trade routes into Europe and moved from trade routes again into India and into China. We just don't have the same records, and because we're a Western country, we think more about the bubonic plague affecting Europe, but it affected the whole world. So any questions up to there? Let's take a break then. Oh yes, you have a question.
This is also a closed system?
This is also a closed system in that it's a series of tubes, and once it gets inside the tubes, that's a closed system. And that will then drain in a sort of a low pressure way back into the subclavian vein.
Is there any sort of like interplay between the systemic vasculature and the lymphatic system that changes like certain concentrations of iron that can alter the concentration of blood in any sort of way?
I don't know that there's an interaction between the lymph and the blood that's regulating ionic concentrations, for example. Now there is a connection in that unlike the cardiovascular system, which has an intrinsic pump, the lymphatic system doesn't have an intrinsic pump. And the way we move fluid through the lymphatic systems is commonly to have those lymphatic trunks associated with the large vessels like the aorta. And it's the movement of the aorta, which now, with each beat of the heart, which passively moves the lymphatic trunks, which is thought to move lymph from the periphery back to the center and into the subclavian vein. So there is that interaction in that you need to have the cardiovascular system moving blood through it, and the actions of the heart and the ancillary actions of the large arteries to move the fluid through the lymphatic vessels, because there is no intrinsic pump. So movement through that system is fairly slow. Yeah.
Yeah, I've heard at one point that it's like the movement of our body helps that.
Right. That's the other thing, is a contraction of skeletal muscle will also help move some of that movement. So again, no pump, so we have to have other means of moving it through. And it's one of the reasons why if you're bedridden for a while and you're not moving, you'll get swelling in your hands and feet. I think it's the reason why if you're hiking and you don't have poles or something and you just left your hands by your side, that after half an hour, 45 minutes, you may notice that your hands start to swell. And that's because you're not moving, you're just sort of, you're not using the muscles of your arms so much when you're walking, and so the fluid is building up in those sites. And if you know someone who's undergone surgery, commonly what they'll do in the tissues where they've removed, particularly they've removed lymph nodes, they'll put in drains. And that's because they don't want to have the buildup of fluid. And so typically the fluid is being drained from the extracellular tissues back into the lymphatics. But if you remove lymph nodes and disrupt that movement, then the fluid builds up in the extracellular space.
Is the lymph in all tissues of the body? You said besides the brain.
Except for the brain, we find lymph nodes everywhere. There's even this old story, I like this story, so I'll tell it. This was understood, this idea of lymph building up and causing swelling was known a long time ago. And the British naval surgeons, because commonly what they had to do after battles, they had to remove people's limbs, realized that they were going to get a lot of swelling in the sites where they'd cut the limb and overlap the tissue. And so what they would do, they didn't know anything about polyethylene drains, so then they always aboard these old sailing ships, they had plenty of rope, and so they would take pieces of rope and sew it into the wound, and that would act as a kind of drain to help relieve this excess pressure that was produced by the generation of lymph, and then they no longer had the lymph nodes to return or they no longer had lymph vessels to return it back to the lymph system. So they drain it until that system could heal. Anything else? Okay, well, let's take a break then, and we'll come back and I'll show you some slides, and we'll head our way into the thymus. Now, this first slide is a highly diagrammatic summary of what we've been talking about. And it's showing the different types of lymphoid tissue. And this structure up here, which because of the two-dimensional nature looks like a series of cold sores, is meant to be the adenoids and tonsils, which are in the back of the throat. And then this is the thymus. We're going to talk about that in a moment. Here's the spleen, always located on the left side and tucked in around the stomach. And down here are the Peyer's patches, the gut-associated lymphoid tissues. The next slide is a sort of a pretty slide trying to describe the idea of hemodynamics. And what it's trying to show is here's an arteriole, and these arterioles are branching into capillaries, and these little structures here are meant to be the endothelial cells forming the capillary. And as the pressure that's generated out of the arteriole, if you go all the way back to the left-hand side of the heart, that pressure is forcing fluid out of the blood capillaries. Some of that fluid is moving back into the blood capillaries because the protein's left behind. That's the story of lymph generation. But now we have forced this fluid out of the blood vascular compartment into the extracellular space, and we're bathing all of these cells. Any pathogens that are out there potentially can be picked up by the flow of the fluid from the extracellular space and into the lymph capillaries. The lymph capillaries now are going to carry this lymph through that series of tubes all the way back to the subclavian vein. But en route, the lymph now flows through these structures, the lymph nodes. And again, here are the afferent lymphatic vessels carrying this fluid into the lymph node. And then these darker regions are meant to represent the germinal regions. Those are sites where we cluster together cells of the immune system. So now we have all manner of cells of the immune system that are waiting there to monitor the fluid that's moving through the lymph nodes. And that fluid has previously been in the extracellular space. So any pathogens that you may find in the extracellular space can now be swept into the lymph nodes, and we can use these germinal regions to monitor that fluid. And then this is a sort of a brief summary of the lymph system, and showing these coffee bean-like structures are meant to be the lymph nodes. These little lines are meant to be the lymph vessels. And you can see that they're meant to be saying here that all throughout your body you have these lymph nodes through which the lymph is passing, and it returns to the subclavian veins. The pathways are a little different on the left side than the right side. We're not going to worry about that so much. But those are the general idea of the lymph nodes. And here's a picture of someone having a truly medieval experience. What you're looking at here is a swollen lymph node. These are the buboes. And this guy has bubonic plague. It's not a problem. I mean, he can be given antibiotics and it will go away in a couple of days. They didn't have antibiotics in the 1340s. And so it killed a lot of people. But now we can treat these very easily with antibiotics. And in fact, there's even some bubonic plague in our area. It's carried on fleas that live in the prairie dog communities. And sometimes, now and again, someone will have an animal that's gone out into the prairie dog community, and they'll get the bubonic plague and bring it back to the home. And then a veterinarian will look at the animal and say, it's got the plague, and everybody has to be treated with antibiotics. It's not a serious problem these days unless you breathe it in. There's a form of the plague called pneumonic plague, and when those bacteria come in through the respiratory system and get into the lung, then it's very hard to treat. And there was, years and years ago, there was a fellow whose cat was under his porch, and he crawled under the porch to rescue his cat. His cat wasn't feeling well. His cat sneezed in his face. Turned out the cat had pneumonic plague, had bubonic plague, had pneumonic form, and the guy never recovered. It killed him. And so these things are still a threat, but with antibiotics, not so much. Yes?
So what's the fluid that's inside those lymph nodes now? Isn't there a fluid in there, or?
Oh, yes, what you're actually looking at is the buildup of fluid. You're not looking at the bacteria themselves. What's happened is the bacteria have gotten into the lymph nodes. They've then started to replicate. They've probably helped block the efferent vessel so the fluid doesn't leave. Also, there's increased inflammation. That's also going to increase the amount of fluid in the lymph nodes. And you'll get this now and again. I'm sure at some point in your life you've had a cold or some infection, and someone comes along and feels for the lymph nodes, which are right along the line of your jaw. And you'll have swollen nodes. You'll have swollen lymph nodes. And those swollen lymph nodes are a result of having some kind of a bacterium or pathogen, could be a virus, in there that's causing inflammation. And that causes the swelling. But that's extra fluid.
So what would make it turn black then? Because you say it turns black.
I think that what they turn black when they begin to necrose, when the fluid doesn't move through enough, and then because of the toxins produced by the bacterium, those cells, the cellular components of the lymph nodes and lymph vessels begin to die and turn black.
And then one last question. So why does it affect like prairie dogs, for example, but it affects humans?
I don't know whether or not it affects prairie dogs. I'm not sure we would know if prairie dogs are dying. I think they do. And in fact, I think that was one of the problems with the black-footed ferrets. And black-footed ferrets are partly because we've destroyed the whole ecosystem. The prairie ecosystem is largely gone. And so the prairie dogs, which used to be in these vast colonies, are now shrunk into tiny islands. And so that's one of the reasons why you can't support the predator, the black-footed ferret. Black-footed ferrets were basically adapted to live in the prairie dog towns and eat prairie dogs. And I think one of the things that began to cause the death of the black-footed ferrets was the plague, because they were exposed to the plague. And I think they commonly died of the plague. So whether or not the prairie dogs die of it, I don't know. But rats tended not to die of it as much either. And rats are thought to have been the major vector of the bubonic plague in the Middle Ages. And they would be carrying fleas, and the fleas would then transmit the bubonic plague to the rats, and then the rats, then the fleas would get off the rats and get on people, and then people would get sick. So, but I don't know what the lifespan is of a rat with the bubonic plague, or a prairie dog. I don't know if they can lead normal lives, or if their lives are truncated. I don't know that answer. There are many creatures, like bats. Bats are particularly of interest now because of the coronavirus. And they will carry any number of pathogens. Bats can carry rabies viruses, for example, and doesn't seem to affect them much. And they probably carry any number of these coronaviruses, and it doesn't seem to affect them much. And they live in these colonies, so they can pass these viruses back and forth. And the problem then arises when the virus leaves those colonies and enters the human or other animal host. Those are called zoonotic diseases, and this is and bubonic plague is a classic zoonotic disease. Anything else? Okay, then with that, I want to start talking about the fourth of those lymphatic tissues, which is called the thymus. You may have noticed on that slide it showed the thymus. The function of the thymus is a little different than those other lymphatic tissues. The role of the thymus is to select for T lymphocytes. The T lymphocytes are going to be a part of the immune system, and we're going to see what they do here probably tomorrow. And the T lymphocytes are selected for potency and efficacy in the thymus. naive T cells are born in the red marrow. And these naive T cells migrate to the thymus. Now, there are a whole host of cells in the thymus that are going to do this selection, but I'm going to simplify it a whole lot, and I'm going to make it a bit anthropomorphic, so you can, I hope it helps you understand what's going on here. And these naive T lymphocytes find their way to the thymus, and in the thymus there are epithelial cells. And these epithelial cells are sites where naive T cells bind. And the way I imagine this is that one of these naive T cells comes along, binds to the epithelial cell, and there's a kind of greeting, a kind of handshake. Hello, how are you? I'm a T cell. Hello, I'm an epithelial cell. And then there's a kind of quiz that the epithelial cell asks of the T cell. And the first question is, naive T cell, can you recognize proteins? Do you have proteins on your surface that can recognize some kind of a protein? And if the T cell says, no, I don't have any proteins on my surface that can recognize proteins at all, then the epithelial cell says, you're useless, go die. Now, in fact, what happens is the epithelial cell recognizes the T cell, and if it doesn't recognize transmembrane proteins that will recognize proteins, it denies growth factors to the T cells. That induces the apoptotic machinery in this naive T cell, and it dies. All of our cells have these genes which generate proteins which are part of the apoptotic pathway. It's a kind of self-killing cell. So the cell will kill itself by when it no longer receives growth factors. So basically, we're saying this T cell is ineffective. It lacks efficacy. But then along comes another T cell, another naive T cell, and again, there's the handshake. The epithelial cell says greetings, and the naive T cell says hello. And now, again, the epithelial cell asks the naive T cell, do you have transmembrane proteins on your surface that recognize foreign proteins? And the naive T cell says, yeah, I can recognize foreign proteins. And the epithelial cell says, well, what kind of proteins can you recognize? And the naive T cell says, I'll recognize any protein you throw at me. I don't care. In that case, the epithelial cell says, naive T cell, you recognize any proteins, you'll recognize our own proteins as foreign, you're dangerous, go die, and withdraws growth factors. But the ideal cell is a kind of Goldilocks cell. And when this sort of Goldilocks cell arrives at the epithelial cell, again, there's the handshake. And the epithelial cell says to the naive T cell, do you have proteins on your surface? Do you have transmembrane proteins that can recognize other proteins? And this naive T cell says, yes, I have those kinds of proteins. And so it's effective. But now the question is about its potency. And the epithelial cell asks the naive T cell, okay, you recognize proteins, but what kind of proteins can you recognize? And if this naive T cell says, I only have transmembrane proteins that recognize proteins that are not part of us, non-self proteins. And if this epithelial cell tells the T cell that it can't recognize foreign proteins, or the T cell tells the epithelial cell says it can recognize foreign proteins, but not proteins that are part of your cell system, then that T cell is retained. So in that case, it's selected for efficacy. It can find foreign proteins, but its potency is also modulated so that it's potent only against foreign pathogens and not against self-proteins. Commonly what will happen in the thymus, a mistake is made, and when that mistake is made, you see these T cells, which are meant to be T cells that don't recognize foreign proteins, now recognize foreign proteins. These are T cells that are meant not to recognize proteins that are on our own cells as foreign, but instead recognizes our own proteins as foreign. That's an autoimmune disease, and that's a problem. Yes, you had a question.
So does the red marrow also have a place, sort of like the, what's in the stuff in your mouth, what are those called?
Tonsils?
Tonsils, like how tonsils have the crypts, and they collect the protein.
No. The red bone marrow will have hematopoietic stem cells. And those hematopoietic stem cells will then, in the red bone marrow, can be selected to become erythrocytes, or they can be selected to become macrophages, or they can be selected to become T lymphocytes. If those hematopoietic stem cells see the right kinds of growth factors, the right kinds of signals, right, kind of something we'll call a cytokine, that will then turn on certain genes that will then direct that cell to become a naive T cell. Once it becomes a naive T cell, it still isn't mature. It can't do its job. It has to now leave the red bone marrow, get into the blood, find its way to the thymus. Then in the thymus, it has to interact with these epithelial cells to be selected to be both effective. It needs to be able to recognize protein, but not be so effective that it recognizes our own proteins as foreign. Okay? Does that answer it?
So does it look for other, when it's getting those transmembrane proteins that are not self-transmembrane proteins, where do those come from then? Or
So the proteins that we're going to see on these T cells are going to have a certain name. They're called T cell receptors. And the T cell receptors are going to be the transmembrane proteins that recognize foreign proteins. But there's a certain dance that has to be done. These T cell receptors are only going to recognize proteins that are put on other proteins on other cells called MHC1 and MHC2 proteins. We'll come to that. Anything else? Yeah.
Just to clarify real quick, so the epithelial cells that are doing this whole process are epithelial cells of the thymus?
They're epithelial cells of the thymus. I way oversimplified this. There are going to be these naive T cells are going to migrate from place to place to place. And there are, I don't know how many, but there's several layers of epithelial cells that these have to be passed through. And each step they're selected either for potency or for efficacy. And at some point, if they recognize their own protein, our own proteins, they'll be rejected. Yes?
So when you're saying naive T cells, that's basically a T cell that hasn't been selected yet?
A naive T cell is one that hasn't been selected. It's naive because it has not seen foreign proteins.
Okay, and once it's selected, then it will be a T cell, and if it's not, then it will
Well, you're right in the sense that when it leaves the thymus, it's going to still be a naive T cell. It hasn't seen foreign protein yet. So it will go out into the germinal regions and hang out there waiting to be able to see a foreign protein expressed on another cell. And then if it sees a foreign protein expressed on another cell, then it'll have interactions with other cells and that cell will proliferate. We're going to tell that story mostly from the point of view of B cells. But we haven't even separated our immune system into B cells and T cells yet. But we'll do that. So, but you're right. They come in absolutely green. They come in, they have never seen another protein. Then before they see a protein, though, we select them to be able to see proteins, but not see our own. Then they go out into the world and they wait. They have a few weeks to wait and see if they find some of those foreign proteins. And if they find the foreign proteins, then they'll proliferate. And some of those then will be banked away in your immune system, so the next time you see a foreign protein, you can proliferate those cells in short order. And we'll talk about that too. Does that answer it? Good. Anything else? Okay. So that pretty much brings us to the end of the story of the lymphoid tissues. And now we can drill down and begin talking about the immune system. And we can divide the immune system into these two big categories. One is said to be the innate or non-specific immune system. And the other is said to be the adaptive or specific immune system. This innate or non-specific immune system is kind of a holding action. It's kind of a holding action against pathogens. It's effectively buying time for the specific or adaptive immune system to take over and finish the job. But it may take 10 days to 2 weeks to gin up this system. So in the meantime, we're just trying to hold the pathogen at bay and keep it from overtaking our systems. When we think about this non-specific or innate branch, we first think that it's a rapid response. This is minutes to hours. It's a very rapid response. Second, it's not highly targeted. And in fact, in many cases, it can be destructive. And we're willing to pay this price. We're willing to pay the price of potential destruction by this branch of the immune system because we've got to hold the pathogen at bay before it kills us so that we can use this branch to selectively deal with it. The cells that we see in this branch are particularly the cells called the granulocytes. And there are three types of granulocytes. One are called the neutrophils, and these are the ones we're going to concentrate on. The second are the eosinophils. And the third are the basophils. The neutrophils, we're going to see, can secrete free electrons. And these free electrons will now produce things like hydrogen peroxide and bleach. These are pretty toxic kinds of molecules, but we're willing to entertain these toxic molecules in a local region so that we can fend off a pathogen, usually a bacterium. The eosinophils secrete, again, electrons that turn into bleach and hydrogen peroxide. But the eosinophils are directed at parasitic worms. In our modern world, we don't think of this very much. We don't carry a particular burden of parasitic worms, but you don't have to go very far back. And people commonly had parasitic worms. And what's more is the parasitic worms can be damaging. Typically what happens if you take in a nematode, a parasitic nematode, that the eggs will hatch. They begin to hatch when they reach the stomach. So you eat them on some piece of food. They come into your stomach, and in the stomach there's a flash of acid because of the low pH of the stomach. That signals the nematode, which is partially developed in the egg, to begin developing a little farther. It then, in the small intestine, comes out of its shell, burrows through the wall of the small intestine, gets carried by the hepatic portal system to the liver, then undergoes a molt in the liver, comes out of the liver and goes from there to the lungs. And in the lungs, it burrows through the alveolar spaces of the lungs, and you cough it up and swallow them, and then the nematode grows up to be its adult form. But throughout that process, there's a lot of cellular damage that's done in something called the visceral migrans, as these nematodes migrate through the organs. And so there's a strong selection pressure to stop that migration as best you can. And the eosinophils are what are largely involved with destroying the parasitic worms during that migration. It's a non-specific response. They're not identifying one nematode or another, but they are killing those nematodes as they migrate. Now, many of you probably are aware of eosinophils. If you have allergies and you have a blood draw taken, they'll tell you when you're having your allergies that you have high eosinophil counts. So it's not only a response to nematodes, it's a response to any number of allergens that we can breathe in, particularly into the lung. And there are some people, and this guy did this experiment, I find it astonishing. He had terrible allergies. He had hay fever. He hated his life in the summer. And so he took a chance. What he did is he left England, where it was home, and he went to the tropics, and he marched around the latrines in the West African area. And in this area, they had a high level of hookworm. And what the hookworm does is it burrows in between the fleshy areas between your toes, then migrates through, does that visceral migrans, and ends up, though, in your small intestine, where it basically is a parasitic worm in the small intestine, but it's sucking blood. Hookworm's a little dangerous because they'll actually make you anemic. But he decided that if he could titrate it just right, and he got to the point where I think it was 12 or 13 hookworms, was just right for him, that would in some way suppress his immune response, would suppress the eosinophils, would suppress the allergies, and he no longer had hay fever. He had parasitic worms, but he didn't have hay fever. And he was willing to pay that price because he was so miserable with his summer allergies. But again, we don't know why this is. We don't know why, if you get a parasitic worm infection, that inhibits the immune system. It's something about the nematodes that are inhibiting the immune system, or whether now the immune system is so worried about the parasites that it doesn't worry about some pollen you've breathed in. And we just don't understand what's entirely going on there. But it seems to work. And there's some people who hypothesize that we have so many allergies in our modern populations that this is this is part of the immune system that gears up to do something. It looks around and says, well, I don't have any nematodes to work on, let's work on some pollen. I find that a little far-fetched, but there certainly is this correlation between those people living in a modern society and having large numbers of allergies, and those people who live in societies where they're exposed to nematodes and they don't have allergies. So it'll be interesting to see how that sort of turns out. Finally, the basophils are basically circulating mast cells. They release histamine. But again, what they all have in common, they're all under this heading of granulocytes. And the granulocyte gets its name from the 19th century histologists, where they saw these cells, whether they were eosinophils that stained with eosin, or basophils that stained with basic dyes, or neutrophils that stained with neutrophil dyes. What they all had in common is they were chock full of these, what they called granules, which we now know as vesicles. And those vesicles, through exocytosis, will be releasing electrons, and those electrons then will lead to the production of bleach and hydrogen peroxide. Then on this side of the ledger, The adaptive immune system, this adaptive immune response is slow to begin. In fact, it starts with a single naive cell. That cell has to proliferate. It has to make thousands and thousands of copies of itself. So that takes time. But the other characteristic of this adaptive or specific immune response is that it's highly targeted. That these cells recognize amino acid sequences between 16 and 20 amino acids long. It's recognized in the three-dimensional shape of these short segments of amino acids. So it's very specific. This branch is basically just a cannon. It's blasting anything that's in sight. But this branch is highly targeted. It's going to target 16 to 20 amino acid sequences of pathogens. And the third thing is that it has memory. For this innate response, every time you get an innate response, every time you get a new splinter, or every time you see another virus, it's pretty much the same. It doesn't remember having done that before at all. But this system has memory. And it's the reason why once you generate an immune response, you can be protected for some period of time. There's a lot of argument about how long this period is, particularly with vaccines. But for things like measles, if you've had measles, you have a lifetime immunity to it. If you've had smallpox, you never get it again. It's even some thought that if you've been immunized against smallpox, that because that's a live virus, that you are now permanently immune to that disease. So it has memory in that once it recognizes a foreign pathogen, it can call up that memory system, and we'll see a little bit how that's done. It can call up that memory system and within a matter of hours generate an immune response that's highly selective. We'll see it has a whole bunch of these cells in reserve. It can recruit quickly. So those are the major differences. And the other point I want to make about this adaptive immune response is that, put it up here, and that's the cell types. When we talk about the adaptive immune response, it's broken into two great categories. So we've already broken immunity into adaptive and innate. And the adaptive immune response is going to further be divided. So the adaptive immune response is divided into something known as the humoral branch. And something known as the cellular branch. The cellular branch are the T lymphocytes. So these are the cells that have to be selected in the thymus. And they also come in two varieties. If you take an immune course in immunology, you'll see there's subtypes upon subtypes upon subtypes. But we're going to say these T lymphocytes can be divided into T helper cells and T killer cells. The humoral branch contains one cell type. These are the so-called B lymphocytes. When we look at these T helper cells and T killer cells, what they have in common is they contain T cell receptors that recognize pathogens on other cells. We'll see a little later that these pathogens have to be expressed on something either as an MHC1 protein or MHC2 protein. And we'll get into the weeds on how that gets done. But one of the things I want you to hold on to right now, and I think it'll make it easier when we come around to some of the details, is that these T cell receptors are always transmembrane proteins. When we look at the B lymphocytes, we'll see there's a period of time when they're transmembrane proteins. But in fact, for the most part, B lymphocytes secrete antibody into the extracellular space. And antibodies recognize antigen to form an antigen antibody complex. So that's pretty much the overview of where we're going to be headed. And we'll walk our way through all of these characters. We'll talk about where the characters come from. We'll talk about how they're selected. We'll talk about how they act. We'll talk about that to some extent for the T cell branch, but particularly for the B cell branch, because these are going to be secreting antibodies. All of this, I think, if you hold on to the outlines of this, this will become useful to you when we come tomorrow and the next day into the details of it. But before we go, let me finish by telling the story of interferon. As a nonspecific / innate response to virus. There was a time I told this story with respect to influenza virus, and that seems really passé now. So I'm going to tell the story from this point of view because of something it's more topical to COVID virus. We've dealt with this a little bit before. But we start here with a layer of epithelial cells. And this will be a respiratory epithelium. Typically for the SARS-CoV-2 virus, these are going to be epithelial cells which will be targeted by the virus in your upper respiratory tract. They could go all the way down into your lungs, but they for the most part appear to be in the upper respiratory tract. And on these epithelial cells, there's a transmembrane protein called ACE2. We're going to see later that when we talk about the kidney, there's an ACE1 that's angiotensin converting enzyme. It does something controlling blood pressure. But this is simply a transmembrane protein which we find on the epithelial cells. And all of your cells in the respiratory epithelium contain it. And then along comes one of these coronaviruses. This is going to be the SARS-CoV-2 virus. This is the nemesis right now. Basically what this SARS-CoV virus is an encapsulation of RNA. There are only about 13 genes on that RNA. It's a pretty small structure. But for our discussion today, the character is going to be this protein, this transmembrane protein. It only has 3 transmembrane proteins. An envelope protein, I think it's an M protein, and this protein called the the spike protein. The coronavirus, in fact, gets its name from the spike protein. When they first looked at these, it looked like it had a crown because of the spikes coming out of them. And there are a whole variety of coronaviruses. And there's some that cause colds, there's some that cause diseases in other animals, but this one happens to have been from a bat. It came out of a bat somehow, we don't know quite how, and it then has the ability, the spike protein has this ability to bind to this ACE2 receptor. So the spike protein binds to the ACE2 receptor. That's the first step. That sticks the virus to the cell. So now the virus is stuck to the cell. It's a little unclear what happens next. It's a little unclear if this happens on the surface or whether it happens internally. But I'm going to tell the story from the point of view of how this happens internally. So this is one of these epithelial cells. Here is the ACE2 protein. Here is the spike protein still attached to the virus, attaches to the cell. That signal now appears to tell the receptor, the ACE2 receptor, and the virus to be endocytosed. So we're going to bring this thing into the cell. So through endocytosis it comes in. Then it forms a vesicle with the spike protein bound to the ACE2 receptor. Exactly where the next step occurs isn't quite clear. It's not clear. It might even occur on the surface. It could occur in the vesicles. It could occur in the lysosomes. But what happens is that there's a protein. There's a transmembrane protein. It's one of our self proteins. I'll put it out here. Let me not put it in green. Put it in red. This has a name. It's TMPRSS2. It's transmembrane protease. There's not an R. No, it is an R. Transmembrane protease serine active site. This is a detail. But this is a protease that attacks the spike protein. If we look at the spike protein, there is a region in it called S1 and a region called S2. And in between S1 and S2, there is a cleavage site. That cleavage site is the target for TMPRSS2. So what happens then is that the spike protein, and I'll draw this as though it's not inside the cell, but the spike protein here is attached to the virus. And when it's cleaved at this S1, S2 interface, this S1 region now contains a hydrophobic amino acid sequence. It's that hydrophobic amino acid sequence which allows this virus with its hydrophobic region here to basically penetrate through the vesicle. So it leaves the vesicle because of the hydrophobic amino acids. It opens up the phospholipid bilayer, and now this virus is delivered into the cell. At this point, the virus comes uncoded and the RNA goes out and the RNA begins to replicate itself. It's an exquisite machine. If you read the details of this machine, it's just astonishing. It goes out into the cytoplasm and it begins to replicate. But it not only begins to replicate, it begins to suppress all of the the non-specific, the interferon system of this cell. So it hijacks the cell and then suppresses the immune response, the innate immune response. I'm going to tell you what that immune response is because that's the basic story. But in fact, in this case, we're going to turn it off. It only has about 13 or 14 genes. Maybe it's as many as 20. I think it's 13 or 14 genes making about 20 proteins. And what they do then is they then take over the cell. And they begin to make copies, thousands and thousands and thousands of copies of this virus. The virus proliferates inside the cell. The virus then, these thousands of copies, because the cellular machinery inside this epithelial cell has been co-opted to make the viruses. The viruses go to the surface and they go out and they escape. And then those viruses, those thousands of viruses, are free to move on to the next set of epithelial cells, bind to the ACE2 receptor, and they too proliferate. So you get this logarithmic growth of these viruses. So the first thing we do normally is that these epithelial cells generate something called interferon. The interferon is produced by a virus-infected cell. That cell's a goner. There's nothing you can do about it. It's dead. And so in its dying throes, this cell, because of the infection with the virus, will produce this molecule called interferon. Then all of the neighbors have these interferon receptors. And when the interferon binds to the interferon receptors, this now leads to a change in the metabolism of these cells. The binding of interferon to the interferon receptor of these remaining cells, which presumably have not yet been infected, this slows endocytosis. It slows exocytosis. It slows protein synthesis. This is a pretty serious thing to do. You're telling all the cells in the neighborhood that they should stop making protein, or at least slow it down. Stop bringing other cells into themselves by endocytosis. Stop exocytosis. So you're throttling back the entire metabolism of these neighboring cells so that you could slow down the replication of the viruses. It's a kind of holding action. Because now, in the next 10 to 14 days, you're going to be wanting to mount a specific immune response that will recognize mostly the spike protein, but probably other proteins too, to prevent these viruses from entering other cells. But you've got just a couple of weeks, 10 days to 2 weeks, to try to mount a specific immune response. But in the meantime, you're generating interferon. And this interferon makes you feel terrible. This is why you feel so awful. And this is why you have a fever. This is why you feel sick. And when you get the flu, it's the interferon that makes you feel bad. But it's a defense mechanism in the hopes that your specific immune system will have time to mount a proper response and completely destroy these viruses and keep them from binding to your cells. And that was what we'll talk about next time, is how we're going to generate, well, actually we'll have to do an inflammation response first, and then we'll talk about the specific immune response. So any questions to there then? Well, let's call it enough then, and we'll start tomorrow with inflammation.